Hello, my name is Bhumika and this lesson is going to be about structure of carbohydrates. I am going to do another videos on lessons related to this such as glycolysis and the tricarboxylic acid cycle, so may you as well check them out. Alright, let's begin. Firstly, we've got the monosaccharides. Now before beginning, I want you to know that the simplest structure of a molecular structure of a carbohydrate is referred to as CH2O whole bracket N. I'm sorry, I had to draw it this way, but the simplest structure is referred to as this CH2 whole N, and that simply means that there are twice as much hydrogen as there are oxygen and carbon atoms. Alright, so that was the simplest thing to remember. Now let's begin with the first type, which is the monosaccharides. Now in monosaccharides, these are the simplest units. Of course, they give rise to high carbohydrates by binding together with each other. For the first example, we've got glucose, which is the most common type of carbohydrate. And then we've got galactose, and then we've got fructose. Now let us look closely at the structure of glucose here. We can see that it has got 1, 2, 3, 4, 5, and 6 carbon molecules. Now, you can notice that I have written it as alpha glucose. Why do I call this particular glucose as alpha glucose? Because when the OH groups, they are present opposite to each other, we refer to it as an alpha molecule. Now you can see the OH is here at the 6th carbon, but on the first carbon the OH is at the bottom side. Therefore, they're pointing in opposite directions. In the structure of galactose, we can see that OH on the 6th carbon here, and on the first carbon it is here. Therefore, they're pointing towards each other. So that is the reason why we'll call it beta galactose. We've got another structure here called fructose, and that's about monosaccharides. Now, before moving on to the next type, there is something that you should know about condensation reactions. What is a condensation reaction? The simplest definition for a condensation reaction is when products are formed with release of a water molecule, of an H2O molecule. Now let's say I added one glucose, another glucose. These two H atoms along with an oxygen atom, they bind together to give such a bond. So this oxygen is left, it therefore binds to this glucose in such a way. And this bond is termed as a glycosidic bond or a glycosidic linkage. Okay, now let's move on to the disaccharides. What are the disaccharides? They are the condensation products of monosaccharides. Again, we know what a condensation reaction is. A water molecule is released and therefore a product is formed. So now I have taken here, for instance, I've taken here glucose, okay, and then another glucose. As I told you, water molecule is ejected from here, detached from here, it forms a glycosidic bond, and therefore we've got maltose. Now maltose contains two units, which is a glucose and another glucose. Now let's say I have a glucose here and I add up a galactose molecule here. Now, of course, at the central portion, OH and one H from here, it will be given out in form of water molecule, and therefore a bridge like so is formed and it gives us lactose. Lactose, a very common sugar, also known as the milk sugar, because it's naturally present in milk. So this, as a subunit, we've got here glucose and then we've got galactose. Now another thing at this point that you should know is the hydrolysis reaction. Hydrolysis is, in the simplest terms, addition of water molecule. So if I take lactose here and I add a water molecule in this, what will it yield? It will yield the individual subunits. That means a glucose molecule and a galactose molecule. So, moving on, we've got the third category, which is oligosaccharides. Okay, what is an oligosaccharide? Condensation product of up to 20 monosaccharides is an oligosaccharide. Now, if I have a monosaccharide, I add another monosaccharide in the first one, then it'll form a disaccharide. Now I keep on adding monosaccharides to this disaccharide, then it'll form a condensation product, which is, if it is less than 20 monosaccharides containing 20 bonds, then it will be referred to as an oligosaccharide. For instance, here I have taken maltose. Let's consider this whole area. If I had an H here, then it'll be maltose. Okay, now if in this maltose I add another glucose, then what do we get? We get maltotriose. And because it is more than 2 monosaccharides, it is a oligosaccharide. Now let's see here. We've got a 1,4 glycosidic bond. We know what a glycosidic bond is, but why do we call it an alpha 1,4? Alpha because the OH groups, they point in the opposite directions, and 1,4 because here this is the 4th carbon, and here we've got the 1st carbon. This way. Alright, moving on, we have got polysaccharides. More than 20 monosaccharides join together by a glycosidic linkage, and they form or give rise to polysaccharides. I told you, if there is one unit is a monosaccharide, add one more unit, it'll form a disaccharide. Keep on adding units until 20 bonds are formed, 20 glycosidic bonds are formed, and it'll form an oligosaccharide. After those 20 monosaccharides, if you add 1 or 2 or any amount of number, it'll form a polysaccharide. Okay, now polysaccharides are basically divided into 2 categories: homopolysaccharide and a heteropolysaccharide. Homopolysaccharide, as the word itself explains, says that if all the groups attached here, all the monosaccharides attached here are of the same type. If I say it's glucose, 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 and glucose, then it's a homopolysaccharide. In heteropolysaccharides, if I say the first one is glucose, then I get galactose, and I get glucose, galactose, and glucose, and galactose, then it's a heteropolysaccharide. Now, again, they may be further divided into branched, unbranched, branched, unbranched. For homopolysaccharide, some examples are basically, you can say starch is a homopolysaccharide. Let's look at starch in more depth, because it's one of the most important compounds in nature. And okay, so here we've got starch. Now this structure right here is the most basic structure that you can draw for starch. So you can see a very branched molecule of starch, and each hexagon here represents a glucose molecule. Now, if I look at this segment, a magnified version of this segment, then I will get a glucose here, another glucose here, and then I've got another glucose here. But the difference between this and this bond is that this one right here is an alpha 1,4 glycosidic bond, because it's formed between the first carbon of one glucose here and the 4th carbon of the second glucose. But here we've got an alpha 1,6 glycosidic bond, because we've got a bond formed here at the 6th carbon of the first glucose that we considered, and the first carbon of this glucose. Alright, so if I say that my starch has got a linear packing like so, then I will refer to it as amylose or amylose. But if I say that my starch has got a lot of branching, then I would say that it is amylopectin. So that is about the structure of starch, one of the most important molecules. It's a storage molecule, and it's composed of amylose or amylopectin, like that. That's it for the lesson of structure of carbohydrates. I hope you were able to grasp the simplest details. As I said in the beginning of the tutorial, I will be making another lessons on glycolysis as well as the tricarboxylic acid cycle. I did not include them in this because they were it would have been pretty boring to study all of that in one go. That is why I'm going to do another handmade tutorials onto this. I hope you enjoyed it, and I hope you learned something from it. Thank you so much for watching.